Hello, my name is Justas and I would like to introduce you to the PPI 3D web server. So the idea of PPI 3D web server is to facilitate the search and analysis of protein interactions. What we did, we took all the data from the protein data bank, we found all protein interactions and we calculated their surface properties and clustered all the interaction interfaces and protein-protein and protein nucleic acid binding sites. What you do, you take your protein of interest or a pair of proteins, you input them to our web server, and after a simple sequence-based search, you have a list of identified interactions for your proteins or their homologs. You can analyze the experimental three-dimensional structures, and you can also infer the interaction interfaces according to visualized sequence alignments. The key feature of the PPI 3D web server is clustering that keeps alternative interaction interfaces. Here's an example. So this green protein, depending on which ligand it is bound, it interacts from one or another side of this blue protein. And if you cluster the proteins according to sequence similarity only, one of the interaction interfaces would be lost. What we do, we cluster the interfaces according to both sequence and structure similarity. And here in green, you can see that it allows us keeping the alternative protein-protein interaction modes, but the data redundancy, I mean the structures that are solved multiple times, they are reduced very effectively. In addition to protein-protein interactions, we analyze also protein interactions with nucleic acids, and the clustering also helps in this case. For example, in this tetrameric protein binds to 2 DNA helices, but the interaction interface is always the same. PPI 3D can be used also for template-based modeling of protein complexes. Probably this feature is a little bit obsolete now because of AlphaFold, which allows not only monomeric protein structure prediction, but also modeling of protein complexes. Now let's see a quick demo of the PPI 3D website. So this is the very simple interface. You can search for a PDB entry, but you can search using a pair of proteins. But I would like to try just simple one sequence search. I'll enter the example sequence. I submit search, a few seconds for blast search in our database. And it will find all the protein-protein interactions for the homologs of human DNA ligase. So here we have the summary of results. We have protein-protein, domain-domain, protein-peptide, and protein-nucleic acid interactions. Let's see these ones, the protein-nucleic acids interactions. We can cluster them according, so all the data are pre-clustered. We see that there are different proteins which interact with DNA. We can do a quick summary. So we have, according to this sequence alignment, we have two possible binding modes. And when clicking on the numbers, we go to the interaction details page, where we can see the structure, we can download, we can analyze the interaction interface interactively, or download PyMol. We can do some basic homology modeling, and we can also see what residues are in the interface. everything is sortable, everything is filterable. So we have many lysines here. For example, we can also analyze the contacts or submit the interface for analysis in the Voro contact server, where you can do some even more detailed analysis. And here, from this sequence alignment, you can infer the interface from experimental structure to the structure of your interest. So it's a very simple web server, and in the future we are planning to introduce structure-based search for protein interactions, and we also are planning to make our database of clustered protein complexes downloadable. And in the end, I have to acknowledge my colleagues and funding from Research Council of Lithuania, and thank you for listening.